Video 6, here we go. Plan now is antibiotics. First of all, there are loads of little keywords. If you want to have a little extra time with this, pause the video and go through what you think all these are. No surprise, the answers are going to be on the next slide, so pause now if you want to. So keywords: antimicrobial, something that generally inhibits the growth of bacteria, so stops them from growing. Antiseptics or antimicrobials generally used on a living person from the outside. Let's call it that, on a living person, but from the outside. So antiseptics would go onto a cut, onto a graze, onto something where the skin has just been broken, but you're applying it from the outside to stop the bacterial growth from the outside of the body. Disinfectants, things like bleach that is used on non-living tissue. So if you've got an injury, the last thing you'd want to do is apply the wrong substance to it, because it could actually cause more damage than good in this situation. Antibiotics. Now, these ones can actually go into the body through various routes, either as an infusion or as in a capsule, as a pill form. Probably you've looked at that quite a bit with regards to the penicillin growth and Fleming and the whole idea as to the fungal digestive juices and how they're acting on bacteria. Let's call it that. It's kind of chemical warfare between fungi and bacteria that they're doing. But the good thing is, because they are specifically targeting bacteria and the cell walls of bacteria, eukaryotic cells like our cells, we are animals, we have got no peptidoglycan in cell walls. We don't do that, but bacteria do. So that's a bit more detail at the moment. Will be coming. Broad spectrum versus narrow spectrum antibiotics. The broad spectrum ones go after many gram-positives and gram-negative species. However, the narrow spectrum ones are generally for the gram-positive only. Gram-positive with plenty of peptidoglycan, with a thick layer of peptidoglycan where they're acting there. This is recap of your microbiology unit on the side. Bactericidal, that's now actually killing. Look at the word stemcidal. So homicidals, homicide, suicide, fratricide, if you want to kill your brothers or sisters. Bactericidal, that's the killing bacteria part here. And the last one, bacteriostatic. Static keeps it steady. That is something that stops the multiplication or slows the multiplication rate of bacteria down, hopefully enough to allow the immune system to do the rest of the job. So let's go through the basics of bacteria again. So you should already know we've got a for all bacteria, we've got peptidoglycan or murin in the cell wall. With gram-positives, there was a thick layer of peptidoglycan. Gram-negative, you've had a thin layer of peptidoglycan followed by that lipopolysaccharide layer over the top, that extra fat layer. Now, peptidoglycan, it's an amino acid chain. It's literally a protein chain on the outside, and those protein chains are being cross-linked. So think of loads and loads of spaghetti strings, but super glue between the spaghetti strings, sticking them together. Now that cross-linking is done by an enzyme called transpeptidase. Trans to between and peptidase, forming the peptide bonds there. It makes the bacterial cell wall extra strong. Now, bacteria usually live in a range of different osmotic backgrounds. So it can live, many bacteria can live in an almost distilled water kind of background and an extremely salty kind of background. They can cope with a range of salinities around them, so the different osmotic backgrounds. And the best way to destroy a bacteria is to break or weaken that cell wall. We'll get to the workings of antibiotics in a little bit. That was just a quick reminder of your gram staining. If you want to pause, then that's what it is. Crystal violet for staining the peptidoglycan, then fixing the stain with iodine, alcohol to wash it out, decolorize, in between always a bit of water, saffron in for the counter stain for your gram negative. And your crystal violet, the colors you mentioned is purple or violet, for the saffron in red/red pink. Yeah, that was your gram positive reminder. Remember that in the gram-positive, the crystal violet stain stays in because it's a lovely thick layer of peptidoglycan. Benefit, as you haven't got an extra fatty layer above that cell wall. You don't have that extra lipopolysaccharide layer that gram-negative cells have. Antibiotics like penicillin can actually access that peptidoglycan without being stopped. That's why they're, thank goodness, a little bit more, a little more susceptible to antibiotic attacks. Gram-negative, that was a reminder. You've got a thinner layer of peptidoglycan. However, you've got that extra layer, either lipoproteins or lipopolysaccharides around the outside. The crystal violet stain can't really get in the first place, or leaks out of that thin layer out of peptidoglycan very quickly, and later on gets washed out in the staining process, and the red stays in as the counter stain. Right, there were two different antibiotics. You need to know both types and their functioning. Now, as a bactericidal, kills the bacteria, penicillin's your example, and for the bacteriostatic, tetracycline is your example. So let's go through them. Penicillin itself, huge benefit to that, penetrates through the whole thickness of that peptidoglycan, of that murin layer, and affects all of it. If you have a chance, look at that flowchart that's in the textbook, literally the damaging the enzymes that would do the cross-linking, because they're enzymes as well that break the cross-linking. In many things in nature, you have got an enzyme that produces something and a counter enzyme that does the opposite thing. The ones that are doing the musculoskeletal system, you've got osteoblasts and osteoclasts, which do the opposite jobs of making new bone and breaking bone again. In this case, you've got two enzymes that work. One is forming cross-links, the other one is non-stop breaking those cross-links. So if you're inhibiting the enzyme that would make the cross-links, you're gradually weakening and weakening and weakening those cross-links. The cell wall goes more fragile, more brittle, and suddenly the bacteria that is being bathed in a hypertonic or hypotonic solution will burst open and your bacteria is gone. That's the idea of a bactericidal being killed. The second one, the tetracycline, that one is a broad spectrum antibiotic, and it does not attack at the cell wall level. It attacks at the level where you've got your, I don't know if you recognize it on the side, where you've got this one here is actually an ATS ribosome. Okay, you've got the heavy part and the light part of that protein. It attacks in the protein synthesis machinery. Okay, it's a bacteriostatic, and it has some sort of effect against other eukaryotic infections as well. Now, the problem is many bacteria have started to find a way of pumping tetracycline out of their bodies again, or destroying it as soon as it comes in. Now, key bit of what it does, it binds to ribosomes, the small 30S units. Now, if you have a look, I couldn't find a suitable diagram to have the 70S. So, but it bonds to the 70S ribosome and inhibits protein synthesis at the ribosomes there. Now, in the normal eukaryotic cell, you won't find the ATS. Sorry, in the normal eukaryotic cell, you'll find ATS. However, in the bacterial cells, you'll find the 70S. So therefore, it's going to attack there, and causes bacteria to just not being able to reproduce, just not being able to produce their own protein. Antibiotics have been used for ages. There's evidence from Greek times that they've used blue mold from bread and smeared that into wounds. Now, World War II antibiotics have saved loads of lives, and modern farming, my goodness, every little chick is injected with antibiotics, every little piglet is injected with antibiotics, just because if they were to have an infection, you have got lost livestock, or they were to not grow as well, and you lose money. So spending the money on preventative antibiotics is, for the farmers, more beneficial. Long term, the dilemma of where are we going to get new antibiotics from if bacteria are evolving quicker than we can manufacture them, then we've got a big problem, and that's sadly on the horizon. And antibiotic resistance is happening. Our problem is we've got, let's say, billions of bacteria around. You're subjecting those billions of bacteria with antibiotics. Out of those billions, just one or two of them might be resistant, accidentally, might have the gene that does something to cause them to be resistant. We'll get to that in a second. Now, bacteria can conjugate and exchange plasmids. We've done that in the microbiology unit, which now means that the surviving bacteria can even share their accidental advantage with other bacteria, do well, they grow, and here we go. We have got the perfect superbug being made. Now, two different ways in which they can obviously acquire that antibiotic resistance, either through an accidental mutation. There is a background mutation rate. Bacteria generally don't have a proofreading enzyme. So however they are mutating, they're mutating faster than eukaryotic cells. That's the first way. And the other one is that they've acquired a fancy mutation from another bacteria that caused them to be resistant. Now, genes do code for proteins, you know that already. So if we have got some sort of way to cause antibiotic resistance, so either stopping the penicillin from binding, making your layers a bit more impermeable, finding ways of pumping the actual antibiotic back out of the cell, they're different mechanisms. And there are links to evolution as well. Now, in the wild, you would have a disadvantage of bacteria that have got some sort of antibiotic resistance, because antibiotic resistance only is a benefit if you've actually got antibiotics about. There's otherwise no point in making I don't know, let's call it an antibiotics pump, and bacteria having that antibiotics pump to pump the antibiotic back outwards. There's no point in wasting ATP to produce such a protein. So antibiotics resistance is actually against the normal evolution, but we've given it a new environment and therefore different environmental pressures. Now, the continuous use, on the high use of antibiotics, and that's farming as well, generates that resistance problem. So if you hear of doctors being far more awkward when it comes to, do you get antibiotics for a little sore throat or not? It's now no longer, oh, I've got a sore throat, I need antibiotics. Doctors will actually say, well, then be sick for 2-3 days. If you're not actually at risk of further complications, then being sick for 5 days rather than 3 days is okay. Various bacteria, leprosy, tuberculosis, gonorrhea, show resistance to some way. Obviously, MRSA, the methicillin-resistant Staph aureus, that one is a big problem. Hospital superbug, you might know MRSA as well. CD infection, Clostridium difficile infections, they are digestive issue infections, and it's a bit of a weird way how we're treating them at the moment. There is only one more antibiotic that works against C. difficile. Basically, it causes diarrhea. It causes you to have a digestive tract infection, and it doesn't go by itself. We've found one antibiotic that still works on it. The problem is that antibiotic is also being injected into livestock in farms. So how long it's going to take until there are antibiotic versions of that C. difficile around, then we've got a bigger problem. In absolutely resistant cases, there's now the idea of a poo transplant, that you'd get the bacteria combination, the healthy bacteria from a healthy person, and getting those bacteria, a poo transplant, a stool transplant, fecal transplant is the technical term, that is now being watched. There are a couple of links in case you wanted to watch those from home. It's in the video that I've uploaded, in the PowerPoint I've uploaded as well. Right, if we had chance to do the practical work with different antibiotic discs, we would have seen exclusion zones around specific antibiotics. And that's the sort of testing you would do if you were to have a weird bacterial infection, to find out which antibiotic is it still going to succumb to. So do that in a lab, and then obviously if you've got an antibiotic, prescribe that to the patient.